So welcome to the last lesson in the SL component of the biochemistry special topic. And by the end of the lesson, you should have an appreciation of what xenobiotics is, discussion around plastics, have an appreciation of what host-guest chemistry is, look at enzymes through the lens of their uses, have an appreciation of what biomagnification is, and also an understanding of the basic principles of green chemistry. You should also be able to discuss the increasing problem of xenobiotics, describe the role of starch in biodegradable plastics, apply host-guest chemistry to the removal of particular pollutants, describe an example of biomagnification, and discuss the challenges and criteria in assessing the greenness of a substance for biochemical research, including atom efficiency and economy. So, xenobiotics. So the rapid development of organic chemistry in the 20th century led to the industrial production of pesticides. Lots of drugs and other chemical compounds that had no natural sources and therefore were foreign to living organisms, the xeno in xenobiotics. These compounds were called xenobiotics and are generally toxic to various life forms and are more resistant to biodegradation than naturally occurring organic molecules. Certain xenobiotics, like persistent organic pollutants or POPs, can remain in the soil and in animal fatty tissue for decades after their release into the environment. And one example is Teflon, which is something I'm quite interested in as a POP. So, major issues around xenobiotic problems associated with them anyway is involving the metabolism of xenobiotics, the bioaccumulation. So depending on their chemical structure, some xenobiotics can be completely digested by microorganisms, plants, and animals, including us. However, many synthetic compounds produce toxic metabolites, or when I say metabolites, like products when they break down, alter the metabolic pathways of other compounds and affect, for example, reproduction, development, and the growth of living organisms. Certain xenobiotics cannot be metabolized by existing enzymes in bacteria or in our guts or in our bodies, and either remain within the organism or are excreted unchanged. So that's where I'm interested in Teflon, because they remain essentially with us for life. The nature of its functional groups and the overall polarity of a xenobiotic molecule strongly affects its rate of decomposition and the environment. Polar synthetic chemicals are often soluble in water and quickly get broken down by living organisms or at least undergo photochemical oxidation. In contrast, the non-polar hydrophobic xenobiotics easily pass through the membranes of our cells and tend to accumulate within the cells of our organisms, in the cells of microorganisms, and also in the fat tissues of us humans and other animals. With such compounds are passed along the food chain, their concentrations increase exponentially, and that's known as biomagnification. Okay, biomagnification, which we'll look at a bit further later on in the lesson. Okay, so this is an example here of the bioaccumulation or biomagnification, leading to biomagnification of DDT. So what happens basically is the insecticide DDT, okay, is put into the water to kill malaria. Zooplankton interact with that, bring it into their cells. Then small fish eat the zooplankton, so it's then incorporated into their cells. Large fish do the same, and then the fish-eating birds of prey, like the hawk, end up eating the large fish and dying, because in every step of the process the amount of DDT in the animal is increasing, because one unfortunate side effect is that DDT affects the thickness of the shells of falcons. So it means that you get lots of death of early birds, because they basically don't get protected. Their shells just break. They die. Another example is the bioaccumulation of mercury, the heavy metal. Okay, and this is known as Minamata disease. Okay, so this was occurred in Japan, also known as itchy disease or ouch disease. So essentially, what happens is that it affects the neurological system of people, nerve endings, causes great pain. Also, it can substitute for calcium in the bones, and it means easier breaking of legs, for example, which can be as the mercury exchanges for calcium in the bones. Not particularly nice. Pharmaceutically active compounds are also a major problem. Here is the example of, say, estrogen type compounds which get into the water table, and it's basically this comes from those ladies who take the contraceptive pill. You go to the bathroom, and then it gets into the water supply. This causes problems because when it gets into the water supply, it creates, because it's basically a female sex hormone, it creates more female fish. And if there's too many female fish, not enough male fish, then the fish cannot breed. Another problem is the PCBs, these ones here. So that's also found in, say, plastic water bottles. So it gets into into you through basically drinking or having it leach out into the water. PCBs are found to cause cancer and liver damage in humans. In 1996, for example, 20 years after the ban on PCBs in plastic, a body of a beluga whale discovered in Canada, in St. Lawrence, contained PCBs in excess of 50 milligrams per kilogram, because it's very hard to break down. That means actually that the whale was classified as toxic to the environment and it was basically treated as toxic waste. And again, discussing plastics, a major issue with plastics, the massive gyres in the ocean, they don't break down, it takes thousands and thousands of years. But birds, for example, and other aquatic animals eat these plastics thinking they're food. This obviously weighs them down, you can't digest them, it destroys their digestive systems, and they slowly, painfully die. Not particularly nice. So you also got the amyrylation of xenobiotics, which involves host-guest complexes, biodegradable materials, enzymes, and microorganisms. So, although enzymatic processes are highly selective and efficient, many enzymes are unstable in the environment and show their optimal activity in narrow ranges of pH and temperature, which we've looked at previously. Certain synthetic molecules are free from these limitations and can selectively bind to environmental pollutants. This creates host-guest complexes. These mimic the structures of enzyme substrate complexes, where the synthetic analog of the enzyme, which is known as the host, and the environmental pollutant, the guest, are held together by multiple non-covalent interactions, including van der Waals forces, ionic bonds, and hydrogen bonds. These cause a problem because they form stable complexes between these host and guest molecules, and in the simplest case, the host molecule contains a cavity of a certain size and interacts with the substrate only by van der Waals forces. Such host molecules combine to a broad range of environmental pollutants, but have low selectivity and interact with many substances that fit into their cavity. The presence of the functional groups that form specific hydrogen ionic bonds with the substance increases the selectivity of the host-guest interactions and makes the host molecule more sensitive to pH and temperature. So this actually can be used as an advantage. So if you want to remove a particular pollutant, okay, so for example, microporous solid materials such as zeolites, which are known, well, are basically alumosilicate minerals, basically immobilize pollutants on the surface of a host material, and then they can actually be taken away mechanically from the environment and removed from the environment and incinerated or destroyed in some other fashion. Okay, so there's an example there. Say you've got radioactive cesium everywhere, you can create a host molecule which will bind to the cesium, which you can then take the host molecule and the cesium out of the environment and dispose of it appropriately. So the other aspect is biodegradable materials. Okay, while many traditional plastics have high biologically inert and don't break down, which we saw with the bird example, biodegradable plastics can be digested by microorganisms within a relatively short time, which you can see here from the image. You've got this biodegradable plastic bottle, which is breaking down quite quickly with the environment. These materials can either be composed of renewable biological materials such as starch, which we've looked at previously, or cellulose, which we'll look at, or contain additives that alter the structure of traditional plastics and allow microorganisms to digest hydrocarbon-based polymers. In addition, certain non-biodegradable plastics such as aromatic polyesters can replace aliphatic polyesters, which are obviously a lot more chemically inert, and the advantage is these are less resilient to enzymatic hydrolysis, so break down more quickly. So this bottle here is an example. It is made of polylactic acid, which is able to break down a lot faster than the associated aliphalic polyesters. There we go. So that's an advantage. So for example, starch plastics can be broken down by enzymes, and they are used for a range of products from disposable bags and food packaging to mobile phones and car interiors. And it's basically reducing the impact of carbon, the fossil fuels in the production of plastics. So, green chemistry. In traditional chemistry, the efficiency of synthetic procedure is measured in terms of the product yield and the cost of raw materials, which is probably what you look at from IGCSE. Well, of course, many other factors such as toxicity, energy consumption, amount of waste isn't really considered. A completely different approach is known as green chemistry, which takes into account these environmental impacts on the entire chemical process and encourages a synthetic design that minimizes the use and generation of hazardous chemicals. So common practices in green chemistry are illustrated above on the slide. You've got prevention, atom economy, which expresses the efficiency of a synthetic procedure as the ratio of the mass of isolated target product to a combined mass of all the starting materials, catalysts and solvents used in the reaction. Okay, so for example, if you have a reaction of A plus B going to C, okay. Now, if you don't use a solvent, the yield can be 100%. Okay, and that's the same with a one with a solvent. But the key is that without a solvent, you're not using a solvent, so it's more efficient because obviously the solvent has atoms as well, and maybe the process also produces some sort of byproduct which is toxic. Okay, with the second process, so you've got to consider that as well in terms of your atom efficiency. So the advantage of, well, you're looking at atom economy means that you're saying, well, the solvent is also part of the problem. So if we do a solvent-free reaction, we're increasing the atom economy. Solvents and catalysts basically further reduce the atom efficiency because their constituent atoms do not form a target product and must be disposed of and recycled. So a non-catalyzed process is even more advantageous. Also, less hazardous chemical synthesis. Okay, so that's again the idea of moving potentially a solvent, designing safer chemicals, using safer solvents. So instead of using, say, tetrahydrofuran, using water, designs that are energy efficient. Other examples are using renewable feedstocks as opposed to non-renewable, so not using, for example, fossil fuels. Reduce the derivatization, okay, number of steps, reduces the waste. Using catalysis, hopefully green catalysis. Design for degrading, okay, real-time analysis of pollution prevention, and using inherently safer chemistry for accident prevention. And that's just the reiterating the idea of atom economy. So it's basically the ratio of masses to determine the practical reaction yield. Okay, so a reaction with a target product of C will be much better produced by AB going to C than AB going to D, where D is this awful toxic side product, because you've got less than 100% atom economy. And here is the equation for it. Percentage atom economy equals the mass of the desired product from the equation divided by the total mass of the products from equation times 100, which is one of the core principles of green chemistry.